Okay, and following these two fascinating lectures, I'm very happy to share our recent research on gut microbiome in gastrointestinal cancer and from basic discovery and to the potential clinical application. To first evaluate the correlation between gut microbiome and cancer, especially colorectal cancer, we did the population-based screening to show whether microbiome altered in CRC patients, colorectal cancer patients, as compared with healthy subjects, including the Chinese population, Germany, French, US, and Austria. And based on the population-based screening, we demonstrating that based on this heat map, and also you can see and then the ecological network, and based on the identified bacteria, they are quite different in terms of composition and also the ecological network. And then this is based on the stool samples. And then move on to the tissue biopsy, also the multiple cohort from normal and the paired adenoma, paired colorectal cancer, to see whether and this change of gut microbiome is an early event or consequence. And based on the screening and by this biopsy tissue to identify tissue adhesion bacteria, also identify change of gut microbiome occur at early stage in patients with adenoma. And this change is more profound and then in colorectal cancer. And based on discovery, we want to answer whether this change of gut microbiome in colorectal cancer patients, whether it can contribute to the colorectal cancer initiation and progression. And based on this question, we pulled the stool samples and from the colorectal cancer patients and also the stool samples from healthy subjects and gavage to the germ-free mice without any additional treatment. And in this germ-free mouse model, we're demonstrating that stool from colorectal cancer patients can spontaneously trigger the enhanced cell proliferation, enhance the pro-inflammatory factor expression, and also enhance the pro-oncogenic factor. And based on this observation, and then especially from the germ-free mouse model and with observation, we're demonstrating that the gut microbiome in colorectal cancer patients is toxin. So we have the group of enriched bacteria and the depleted bacteria in colorectal cancer patients based on multiple population. And then based on the enhanced bacteria, choose one example, the Peptostreptococcus anaerobus. And this study, we're demonstrating that you can see, and based on the electron microscope, and these bacteria have active interplay and with the colon cancer cell line, but not somehow it's a silence, it's a quiet, and with the normal epithelial cell. And then we're demonstrating that this bacteria surface protein can bind into the receptor protein, the integrin alpha-2 beta-1 in the host epithelial cell, colon epithelial cell, which can activate the PI3K AKT oncogenic signaling pathway and therefore promote the cell proliferation. At that time, we also performed single-cell sequencing and demonstrating that these bacteria correlate with the suppressed immune microenvironment. And then we further evaluate how these bacteria play a role in suppressing the immunomicroenvironment in colorectal cancer. And my second postdoc fellow, Yali, and who is my PhD student and also postdoc fellow, he focus on multiple mouse model, and then both of them can demonstrate enhance the MDSC and also the number and also the activity. And based on this observation, we put effort to identify mechanism how these bacteria can induce and then MDSC infiltration and also the activation of tumor suppressor immune cell MDSC. So we, from the host, and because the bacteria have a direct interplay with the host, we're demonstrating that the bacteria interplay with the host cell by activating PI3 AKT signaling pathway can further induce activation RKB and therefore release pro-inflammatory cytokines, for example, CXCR1. And CXCR1 can bind into the receptor and CXCR2 to recruit this MDSC cell to the tumor side. We try different model and different cell line. It can just recruit in the tumor cell but cannot activate MDSC. And then we further to identify how these bacteria can activate MDSC. And then we're demonstrating that bacteria produce a protein, LYTC22, binding to MDSC receptor. And this binding can activate this MDSC activation in the tumor side. And therefore, the activation of the MDSC and then recruit to the tumor cell and then can suppress anti-PD-1 therapy. Based on this observation, we want to block two receptors to see what's happened. We block the host connection and also block the bacteria produce the protein and binding to the MDSC, and then reversed the resistance to sensitive for anti-PD-1 therapy. So this is one example. We identify the pathogen that can induce, promote colorectal tumorigenesis. And then we move on to the gastric cancer because the gut microbiome closely correlated with the gastrointestinal cancer. And the recent study have demonstrated that screening of H. pylori alone is not sufficient to prevent the incidence or reduce the mortality of gastric cancer. Another study also showed that the long-term follow-up for 14 years, community-wide H. pylori eradication only just slightly reduced the risk of gastric cancer incidence in high-risk population. Our question is that we need to identify additional bacteria in gastric cancer. We also do the multiple population to see the change of microbiome and based on the biopsy tissue from superficial gastritis, precancerous lesion, atrophic gastritis, intestinal metaplasia, and also gastric cancer paired biopsy. And based on the screening, the bacterial screening from the tissue sample, we identified Streptococcus and robust as the top enriched bacteria in cancer tissue as compared with the non-cancer tissue in the stomach. And another study, because we have in the high-risk area, we do the random H. pylori eradication. We want to see, after H. pylori eradication, if there is any other bacteria can contribute the persistent progression of precancerous lesion. And this study, we follow up of 1 year. So, and this is from the subject, the high-risk population-based study. We have a random H. pylori positive cases. We randomly treat with a placebo and treat with the antibiotic eradication therapy. And then we screen the baseline after 1 year follow-up with or without H. pylori eradication to see the bacteria that correlate with the persistent progression of the disease, especially precancerous lesion. We do identify to see, you can see the red color, and then there's a group of bacteria that correlate persistent atrophy, intestinal metaplasia. And then we overlap this precancerous lesion, persistent progression after H. pylori eradication, overlap with the enriched bacteria in the stomach. You can see, and this overlap bacteria again, Streptococcus and robust occur again as one of the bacteria correlated with the persistent progression of the precancerous lesion, atrophy, intestinal metaplasia, and also chronic gastritis. We have the question, what is bacteria for as angionosis? And based on the literature review, is this bacteria just caused, and based on name, that caused the infection? The correlation of these bacteria in cancer is totally unknown. We have the question, because H. pylori just a trigger factor induce acute chronic inflammation. One change the precancerous lesion due to the enhanced pH level, bacteria will left. So what happened for this one? We do the different time point at the 2 weeks post-infection. We choose H. pylori as a positive control. It can induce acute inflammation. All the histology evaluation was done by our 2 senior pathologists in our hospital. And then what's happened without any treatment, only this bacteria long-term treatment. So with the conventional mouse model or germ-free mouse model, the long-term treatment 6 months, 9 months, 12 months, we demonstrating that these bacteria alone can spontaneously cause chronic gastritis, atrophy, metaplasia, and dysplasia. And then only this alone is not only an early trigger effect, but the persistent all the progression from the gastritis to precancerous lesion. And also these bacteria can promote gastric cancer in multiple mouse models. And based on this observation, we spent some time to evaluate the mechanism of how these bacteria interplay with the gastric epithelial cell to promote initiation, precancerous lesion, and promote gastric carcinogenesis. We identified bacterial surface protein TMPC binding to the receptor anectin A2 and trigger oncogenic signaling pathway, and summarized in this picture at the acute situation, and this binding to produce a pro-inflammatory factor, but the long-term infection can promote MAPK, JNK oncogenic signaling pathway. If we knock out the TMPC, this protein, surface protein from this bacteria, and this effect will totally abolish. So confirm, and this is a new pathogen, and contributing to the initiation, the whole stages progression of gastric carcinogenesis. So let's move on to the potential clinical application. And based on the microbiome research, the first one, whether the depleted bacteria, are they good, are they potentially protective? And this is based on the multi-population-based study, Asia, and also the Western people, because we want to identify a common feature that correlates with the disease, colorectal cancer. This is the depleted bacteria. We set the mouse model and provided this bacteria can significantly prevent the colorectal cancer progression. So, and then we identify this bacteria produce the enzyme beta-galactosidase, and then this is a functional enzyme. If we knock out the enzyme-related gene, lactate gene, and then the protective function will be gone. We also evaluate the molecular mechanism of how this bacteria produced beta-galactosidase, contributing to the suppression of colorectal cancer is mainly by suppressed Warburg effect. And then we move on to second, the candidate, depleted bacteria. And this by depleted just in female colorectal cancer patients, but not in male. And then based on the mouse model, demonstrating that this bacteria only suppress the colon cancer in female mice, but not in male mice. And based on these multiple observation, and we did a mechanistic investigation, and we demonstrating that this bacteria attached to the colonocyte, the receptor is estrogen dependent. The SLC3A2 is a receptor in the colonocyte and from the intestine, and then with the receptor, and then this bacteria can colonize and stay and produce, harmful metabolite 7-DHC, and further and transfer to vitamin D to protect against the colorectal cancer. So the protective bacteria have a gender difference. And this bacteria, good bacteria package, and then we transfer to a local company, my colleague, and then open a company, and they are working on the clinical trial. They are finished the year 1 clinical trial, focused on the adenoma patients after removal of the adenoma under endoscopy to see whether produce these probiotics to suppress the relapse of colorectal polyps or adenoma, and long-term whether it can prevent the colorectal cancer. But not now. Right now, it's just a short-term clinical trial. They finished the year 1, I already published the paper, and this probiotic formula can suppress the colorectal cancer in enriched bad bacteria, pathogenic bacteria. Another bacteria, and this is this unpublished data, based on the population-based large sample size, and this bacteria particularly depleted in gastric cancer. So we want to identify gastric cancer probiotics as well, and also this bacteria based on the network analysis cohort, a larger cohort, and these bacteria have a negative interplay with H. pylori. And based on this initial evaluation, we demonstrating that this bacteria can suppress H. pylori infection, no matter it's SS strain, the common strain, or antibiotics resistant H. pylori strain. They successfully suppress and this bacteria, the H. pylori growth, and also the H. pylori infection. And based on in vitro study and in vivo mouse model, further we demonstrating that this bacteria can also suppress gastric carcinogenesis. And we have the different mechanistic investigation mainly, and this bacteria can produce butyrate. And that number one, this bacteria can compete the common receptor, which H. pylori. The common receptor is ENO1. This is step 1. And step 2, this bacteria produce butyrate. And this butyrate can suppress the H. pylori, inside H. pylori ATP synthesis, and therefore suppress H. pylori growth. Number 3, and this bacteria produce butyrate, can suppress oncogenic signaling pathway, Wnt beta signaling pathway, therefore, and suppress gastrocarcinogenesis. So we move on to number 2, and then the potential biomarker for microbiome. You can still remember under the heat map the different abundance of gut microbiome in cancer patients as compared with the normal subjects. So we pinpoint and these important or major player as a biomarker for colorectal cancer or adenoma. So we treating F1 is Fusobacterium nucleatum, another bacteria game name M3 because it's a new one. M3 is good for adenoma. Fusobacterium nucleatum is good for colorectal cancer. We have the 4 bacteria, package, and this package compared with the FIT, they have a good performance. It's better to early stage of colorectal cancer as compared to FIT. You can see the blue column as compared with the green column. They have a significant difference. And also, this we listen to our colleagues' company, and they developed the M3 CRC detection kit, now apply in the clinical side in Hong Kong and some Asia-Pacific region. And we continue to look up whether we can also, because in the clinical side, you ask the patient to contribute the stool sample is somehow not that easy. You ask them to contribute the blood, somehow it's quite easy. So we want to see whether we can identify the metabolic panel as a diagnostic marker for colorectal cancer. We do a multiple population, 1 discovery cohort, 3 validation cohort, in total over 1,000 subjects. We did the shotgun mechanism sequencing for stool and the metabolomic assay in stool and the plasma, and also cytokine profiling. And then the plasma metabolomic have a good performance as compared with the stool. So I just show the plasma metabolomic feature in normal control, and then in the middle is adenoma and the colorectal cancer. You see there is a shipment from normal to cancer, increased or decline metabolites according to disease progression. And based on this observation, we identified the metabolic panel. It can significantly distinguish colorectal cancer versus control, early colorectal cancer versus control, and also colorectal cancer versus adenoma. We compare to the FIT, and these plasma metabolic panel have a good performance to distinguish the colorectal cancer, especially early, and as compared to control subjects. And this as a cover story, and just a sample publication in last year. And this is a biomarker, and working with the company, and hopefully to translate. And before that, we have the multi-cohort validation to confirm, to prove, and this plasma metabolomic marker is good and for the potential clinical utility. Finally, I would like to briefly introduce the modulation of the gut microbiome metabolites in immunotherapy. So in colorectal cancer, not like other kind of cancer, is the opportunity to use anti-PD-1 inhibitor and the checkpoint inhibitor is not optimum. It's just 15% or 10% is suitable and for the anti-PD-1 therapy. So we want to see whether the change gut microbiome metabolites can change immune cold tumor to immune hot tumor. And Max, my postdoc fellow, we focus on the Clostridium butylicum. It's a depleted bacteria in colorectal cancer patients. And then we want to see, and because based on the pan-cancer analysis, we're demonstrating that these C. butylicum have a positive correlation with the responders in other cancer types, melanoma, liver cancer, renal cancer, and also the lung cancer. So we want to see whether this bacteria, because the information on colorectal cancer is not available, and based on the pan-cancer analysis correlated with the immunotherapy, we set the different mouse model and demonstrating that these bacteria do and suppress the tumorigenesis and improve the anti-PD-1 efficacy in multiple mouse model, and especially in MSS mouse model is the anti-PD-1 resistance mouse model. So, and based on this observation, we're demonstrating that how these bacteria play a role in increase the efficacy of anti-PD-1 therapy. And based on the mouse model, we also consider whether these bacteria produce the function to increase the anti-PD-1 efficacy is a potential clinical impact. And then we perform the humanized mouse model and prove that these bacteria, say Butyric, can inhibit the colorectal cancer by enhancing the CD4 T cell activity in this humanized mouse model. Again, because we want to consider it's more close to the clinical side. And we have the organoid from colorectal cancer patients. We also have the autologous tumor infiltrating T cell, the TILs, from the same patient. We co-culture C. butyricum with TILs and then treat. And these the same patient organoid. And we have the repeated experiments. We select MSS and MSI, immunosensitive, immunoresistant colorectal cancer. Both, say, butyricum can activate TILs to enhance the anti-PD-1 efficacy and with a significant induction of CD8 port T cell. And this, we summarize the mechanistic observation. The bacteria itself, the surface protein SAD, can bind to the colorectal cancer surface receptor GRP78, and which one can reduce the secretion of interleukin-6, and therefore block the that can enhance the CD4 T cell activity and then induce tumors associated. And this leading to the reduction interleukin-6 and increased activity of CD4 T cell. And this number one. Number two, we did the humanized mouse model and organoid together with the TILs mouse model to prove and these bacteria have a potential application in the future for possible the screening and in clinical as an adjuvant treatment in the clinical side. So, and based on this study, we also identified a few other bacteria can trigger the anti-PD-1, the boost anti-PD-1 therapy by trigger the CD4 T cell activity or suppress the Treg activity or suppress MDSC activity. They can change the immune cold colorectal cancer to immune hot colorectal cancer. Our study also summarized in some invited review articles and then to summarize our discoveries and discovery from outside from other scientists in terms of the basic research or translational research. And based on this invited review article, we can recognize that the study for microbiome is a shift from the basic interplay investigation, mechanism investigation, to potential clinical application for prevention therapy or targeted therapy or adjuvant therapy. So I would like to thank my team members, and some of them are sitting here in this session for their particular hard working and particular contribution to the individual project. I also would like to thank the clinicians and scientists from the Institute of Digestive Disease and the State Laboratory of Digestive Disease. Thank you so much.